Hello, everybody. I first of all want to thank the COX-T study group for committee for giving me this opportunity to present my work, basically our work today. I'm relatively new to the COX-T field, so it's a great pleasure to be here among some of the great pioneers who've been working in COX-T for a long time. And the work that I'm going to be presenting today is a collaboration with one such team at NAU, and I'm excited to be here, and I look forward to learn more, increase connection and collaboration. Okay. So just a brief background, valley fever, as you all know, is a lung disease that's caused by inhalation of aerosolized spores of two different strains, Imitis and Posidacea. Historically, the CDC data basically shows globally valley fever is more prevalent in southwest region of US, Mexico, and some regions of Central and South America. And with the changes in global warming and changes in the weather, is going to be kind of spreading more in the territory. So valley fever mostly is a benign disease because about 60% of the cases are asymptomatic, and 40% that are infected have some symptoms, and of which 5% get severe disease. So the diagnosis of valley fever becomes really important because there are few tests that are available for diagnosis. Like you can culture the fungal directly, but it's a little bit cumbersome, and sometimes the patient does not produce enough sputum and then the sensitivity of those culture is kind of cumbersome, and also the sensitivity is low. The most commonly used, and the initial point of detection is between 1 to 4 weeks of the illness, and the primary antibody that is detected is of IgM class. Serology is the most commonly used way of diagnosing valley fever, and it's detectable by immunodiffusion and complement fixation, which starts normally between 21 to 35 days of illness, and it's peaked around 21 to 70 days, kind of reaching the maximum. And then by almost approximately 170 to 300 days post-illness, the titers kind of fade off. And by almost close to a year, it's very hard to detect any antibody response against valley fever. And as we know that valley fever is a common cause of community-acquired pneumonia, and it's more prevalent in individuals that are immunocompromised or immunosuppressed. And for that reason, in the endemic area, patient that has allergy, autoimmunity, or undergoing cancer immunotherapy and organ transplant, they need to be screened for past infection. And as you can see, serology is normally not able to, because the antibody titers tend to fade away, it's very hard to detect any past infection. So there is skin test. It's a delayed type hypersensitivity response. It's being used, but it's not very frequently used in practice. So basically, that's where our story for the flow assay started. There are a lot of studies in animal model and in human that showed that T-cell responses play a role in control of valley fever. And in addition, there was a study done by Dr. Ampel's group where they showed that whole blood from valley fever, when stimulated with complex antigen T27, it showed an increase in cytokine like interferon gamma and TNF alpha. So we wanted to see if T27 could stimulate these cytokines from PBMCs of valley fever subjects. And as you can see that both CD4 and CD8 T cells in valley fever subject showed an increase in TNF alpha and IL-2 responses compared to healthy control. But surprisingly, we did not see any change in interferon gamma. So the rationale of our study is that although serology is useful for detecting acute valley fever infection, the antibody responses kind of fade away approximately after 300 days. Then our data is kind of showing us that the interferon gamma results are inconsistent with complex antigen. So basically, there's a need for much more standardized and reproducible set of reagents and assay to be able to detect the past valley fever infection. So the goal of our collaborative group is to generate a relevant peptide pool for a more reliable response. And we were able to generate such a peptide pool due to NAU initiative that was supported by U19. And we call it a NAU 108 pool peptide. And we have used this peptide for our further studies to develop flow-based assay to detect memory T cell responses. Okay, so I'll briefly go through the method. Anybody who needs more detail on the method, I'll defer it to Eric Settles at NAU because their team has worked on more details of this process. So basically, two sets were done for protein to identify a 79 peptide library, and then another set to identify 29 selected peptide library. These two libraries were basically pooled into a 108 peptide, and then these were stimulated with valley fever PBMCs, frozen valley fever PBMC, and basically we did the supernatant were analyzed for cytokines by Luminex, and the cells were then processed for flow cytometry assay. So in the flow cytometry, we basically targeted on looking at the memory T cells, activated memory T cells that were secreting intercellular cytokines. So the activation markers are CD69 and CD137. And I do want to emphasize over here that the presence of co-expression of CD69, CD137, along with interferon gamma, indicate expression of specific CD4 or CD8 antigen-specific T cells. So I just want to go through the demographics of the data that we use for our study. So basically, we categorize our subject, non-endemic healthy control. Endemic healthy control were basically from Arizona, and the valley fever subject were from Arizona and California. So we had about 8 subjects for the non-endemic healthy control, 3 for endemic healthy control, and about 13 for valley fever. Now, the valley fever subject were mostly, I think 12 out of 13 were mostly uncomplicated pulmonary category, and there was only one subject that was category 3, which was complicated pulmonary disease. Most of the age group was between 40 to 52 for all the 3 categories, and mostly the non-endemic and endemic healthy control had females. There was better distribution of a male and female and valley fever subjects. I just wanted to highlight that the years from the diagnosis of those subjects to when we did the flow assay varied from about 6 months to 3 years. So going to the result, and I apologize if you're not able to see it very clearly. So basically, we saw that the endemic healthy control showed a significant increase in interferon gamma secretion in the CD4 memory T cell compared to the non-endemic healthy control. And similarly, the valley fever showed an increase, a significant increase in interferon gamma secretion in memory CD4 T cell compared to the non-endemic healthy control. And then when we looked at the co-expression of CD137, CD69, there was a significant increase in valley fever CD4 memory T cells co-expressing expressing these 2 markers compared to the non-endemic healthy control. Similarly, the CD8 memory T cells only showed significant increase in interferon gamma secretion and the endemic healthy control compared to the non-endemic. We did not see much significant change in the valley fever. And then the co-expression of CD137 and CD69 on the CD8 memory T cell was significantly higher in the valley fever subject compared to the non-endemic healthy control. Then looking at the cytokines from the supernatant, we did not see any difference in the interferon gamma secretion for the valley fever or the endemic healthy control compared to the non-endemic healthy control. But there was a significant increase in the valley fever, in the secretion of IL-2 in the valley fever subject. And then the IL-10 was actually lower in the valley fever subject compared to the non-endemic healthy control. So to summarize the data and the future direction, data suggests that NAU108 peptides accumulation and flow assay can differentiate between non-endemic healthy control and endemic healthy control and valley fever patient. We definitely need to further validate our assay related to interferon gamma secretion, expression of CD69, 137 surface marker. We haven't tested any other inflammatory cytokine like IL-17, so that needs to be done as well. And potentially this could be used for diagnostic and could be easily translated into CLIA flow lab, but obviously it needs a lot more work. So based on the subject demographic data and the assay, the flow assay could be used to improve risk stratification, inform prophylaxis strategies, and define eligible individual for future vaccination trial. Our collaborative team is also working on another assay platform, ELISpot, and please see the poster number 43 if you want to get more details on that. And with that, I really want to thank a big collaborative group for us to be able to do this work. Thank you so much.
There we go. All right, we have time for some questions.
Wonderful talk. Thank you very much for that. How do you explain lower level of response in patients versus endemic healthy controls?
So you mean the lower level of response in the endemic healthy control compared to the non-endemic? I didn't get your question.
No. Your interferon gamma response was lower in all the classes, all the diagrams you had on the valley fever patients versus endemic healthy controls.
So if you look at the interferon gamma responses, let me go back. You mean this first one, right? Yeah, so the endemic healthy control responses are it's definitely significantly higher than the non-endemic control. The reason for it, you mean it's lower?
In compared to the patients.
Compared to the patient, yeah. So in compared to the patient, there is a lot of variability from patient to patient. And as you can see, there are some patients that shows a much more increased response to interferon gamma compared to the endemic healthy control, but there are some of them that are lower. And it's just individual subject to subject variability. And I think when we have a much bigger sample size, we should be able to, get a better differentiation. In addition to that, we haven't really looked at individually at each of these patients' clinical data to be able to compare the reason for this difference in the response. And I think that's what we want to look, going forward, look at that, like what could be the reason that some of the endemic healthy controls are responding much more strongly than the valley fever subjects. Thank you.
Hi, I was wondering if you looked at exhaustion markers in CD4 and CD8 T cells, or if you looked at other possible gamma-producing cells like ILCs.
Can you repeat your question again?
Yes, I was wondering if if you looked at exhaustion markers on T cells, or if you looked at innate lymphoid cells in these samples.
No, we haven't. Thanks for that question. No, we haven't really looked at the exhaustion markers on these, but I think that's a good thing to kind of keep in mind for our future studies, because that'll answer a lot of things in terms of the differences in responses for a particular set of patients. Because if you look at some patients are responding higher and the others not, so that's a good point. We'll definitely keep that in mind going forward. Thank you so much.
Okay, thank you, Dr. Kala. And we're nearly on time. We are over, as Dr. Galgiani likes to say, it's not a Coxie study group unless we're running late. Our next talk is going to be from Ana Fabio Braga. Does coccidioides affect the lung microbiome in small mammals? And this is from Pathogen and Microbiome Institute at NAU.
Thank you. We're shooting for 12?